a consultant in life sciences. And today on the podcast, we're going to be speaking with David Baram, and we're going to talk about his company. But before we hop into the podcast interview today, I just wanted to quickly mention I'm headed off to Bio June 5th through the 7th. I will be cutting my trip a little bit short, but I will be there for the first couple days just doing interviews. So for those of you who are around and would like to be on the Life Science Success podcast, please be sure to go to the web address that's mentioned at the very bottom of the screen here. So it's web.d3digitalmedia.com/bio-2023. So with that, in this episode of the Life Science Success Podcast, my guest is David Baram. David is the co-founder and CEO of Emendo Biotherapeutics, and David has taken Emendo from pre-seed stage through multiple financing rounds and successful acquisition in 2020. So with that, welcome, David.
Thank you, Don. Thank you for having me in this podcast. Looking forward.
Yeah, thanks so much. So could you tell the listeners just a little bit about yourself?
Sure. So I think that before anything, I'm a scientist by training, and science has been and continues to be my passion. Before establishing Emendo and while taking the role of the CEO, I think that the scientific angle and the wonderful things that you can do with it, that has always been my passion. I have a PhD from the Weizmann Institute of Science and Structural Biology. Actually, my professor from back then is Ada Yonath. She's Nobel laureate for 2009. My expertise are around protein engineering and structural biology, and a lot of what Emendo is based on that. But in addition to loving science, I think that since childhood I've always been entrepreneurial. I think that I founded my first company before completing my PhD, and Emendo was probably the fourth. So that has always been my passion as well, and that's why I find myself doing what I do, and not in academia, for instance.
Yeah. I mean, your background's very interesting as well, because I mean, as I looked back through, it looked to me like you had like a finance or investment kind of background as well. You work for companies in that space too.
Right. So that's also an interesting development, because when I completed my PhD, I had to make a decision whether I stay with science and I stay in academia and I go and do a couple of postdocs and take a position, or I go and do something else. Back at that time, and that's also because of the research that I held, during my PhD, was very, very high resolution, atomic level questions that I had to answer. And I have always been interested in the broader picture. I wanted to see and feel the applications. And in addition to that, I was, I had like fatigue from doing the actual hands-on research, and being, as I said, entrepreneurial. I said, okay, so how can I enjoy both science and business and being entrepreneurial? And then I turned into founding, together with a partner, a small investment firm. And it was great fun, and it was extremely interesting, and you get to cover a lot of technologies, and you see everything that is going on around. But at some point, I think it was after 6 years of doing that, I miss the hands-on ownership, to be responsible for, making something, building it, creating value, not just supporting the value creation, actually doing it with your own hands, leading the people. So that got me back from investments um, to actually, building startup companies and taking them forward.
Yeah, it's I mean it's an understandable background in your journey. I mean, I definitely as I was going through school, I definitely liked the learning process and kind of going through everything that we had to go through to establish my PhD. But in the end, I was ready to be done as well. And you know, being an entrepreneur now, you know, and having responsibility for everything, you know, is something that you know is a drastic difference than working from somebody for somebody as well.
Yeah, I completely agree with that. And I also think that, I think that when you're an entrepreneur and when you're a leading entrepreneur that eventually takes the CEO seats and you're practically responsible for everything. Uh, Being able to connect all dots into something that makes sense and then executing it, that's something that I love. And I
Hmm.
think that I have found my sweet spot actually. Uh, so I'm very happy about that. Like, because eventually your career can take you to so many directions. But although I have been doing, you know, different things, I think that eventually they led me exact to where exactly I need to be. And it's good. I believe that it's good for everyone when you're able to navigate your career to that exact place. That means that you're better in what you do, you're better for yourself, and you're better for the investors, and you're better for anyone that follows you.
Yeah, that's great. That's great advice actually for anybody that's listening as well in terms of finding that it's not just finding your passion, but finding the spot that you belong more or less as well.
I agree. And actually, sometimes before you experience that, and that's also something that I'm very happy about. I thought it's, being an investor is actually, when you come from scientific work, you go to you go to being an investor, it's glamorous. You think that it's wonderful. It's probably, the best development in your career. And you learn from that and you say, okay, that's nice, but it's not enough. So also experiencing not being afraid to take bold moves, because the easy move for me post my PhD, and I had a pretty successful one, was to go and have my postdoc and continue with that path. I had the opportunities. Taking the different path that takes me to, to being an investor is not a trivial thing to do, but it was the right thing to do.
Yeah,
I learned a lot, and it brought me back to exactly where I need to be. So
that's
taking bold moves and changing and testing it, learning from it, you can always change back.
that's great advice. So let's talk about Amendo for a minute. So where does the name come from?
So Emendo is practically in Latin, it's amending. And since Emendo, you know, amends the DNA in some ways, and along the way, of course, many other things, we thought it was a good choice of a name.
Yeah. Yeah, and I mean, especially given the company. I mean, this is where I'm going to make the pivot to talking a little bit more about Amendo. But I think, to me, you know, looking at what Amendo is doing is very important as well. And so, would you share just a little bit of, you know, the background of the company and what is it that you're working on?
Uh, yeah, so let me start from the very beginning, because the start was, I think it had a major part in everything that we are now. And Emendo was founded 2015, late 2015. So it was, I don't know, a year and a half after the first generation CRISPR companies were founded. Intellia, CRISPR Therapeutics, Editas. I think around 3 years after Jennifer Doudna's and Emmanuel Charpentier's discovery that CRISPR works well in mammalian cells, or could work in mammalian cells. And you know, since the discovery, I found it fascinating. And when the first companies was founded back on based on that technology, I said, "Whoa, like it's actually happening." So uh, we are actually going to manipulate DNA of live human beings in order to cure them. That was fascinating for me, and that's what attracted me there. But then, where do you come in? Because the technology is there, right? There are companies who are already developing it, but it's very early. The technology was so promising that it made a really fast move towards becoming companies. And then these companies went public at very early stages of not even preclinical work, like in vitro work,
Wow.
um, just because of the promise, right? So I was there together with my co-founders, and I say, okay, so that something great is happening here. How can we become part of it? And the thing that attracted me, and I think all of us, to gene editing generally and CRISPR gene editing specifically, is being able to cure people. I think that this is like transformative times in healthcare generally. It will have a huge effect because it's a conceptual change from mainly symptomatic treatments to actually going to the source of the disease and being able to fix it and then cure people. That was the motivation. Now we had to think, okay, so that there's this wonderful technology, but eventually it will be successful if it gets to as many people as possible, if it cures as many diseases as possible. Now we started working with CRISPR just to see how it works, and the initial promise that I thought of said, "Okay, this is amazing. It's like magic. You have this protein element with an RNA element. It is a kind of a cassette that will effectively target any place along the genome, which is an amazing promise. But then biology and reality together knock on your door, and they show you that never works exactly as you anticipate it to be. And that's something actually that didn't change since inception. We said,
Mm,
okay, we want to bring it to as many people as possible. First thing that we want to deal with is safety. We remembered what happened with gene therapy back in the 90s, right? In
right.
1990s. It was tragic, it was dramatic, and it was due to off-target effects,
Hmm.
right? So we said, okay, if you want to bring it to as many patients as possible, you have to make it as safe as you can possibly get it to be. So the first objective was, oh, okay, we'll take CRISPR gene editing, and we'll do everything that can be done in order to potentially eliminate off-target altogether. I'm not talking about just making it less or making it tolerable. We will aspire to eliminate it. This is back at the time, I can tell you, even our investors, our Series A investors were OrbiMed and Takeda. And they were saying, whoa, whoa, sure you want to say that? Alex, you're going to eliminate. And we say, listen, I don't know if we'll get there, but that's what we all of us in gene editing must aspire to. Because
Hmm.
that's like you want to make patients better. You don't want to take any unnecessary risks. So that was goal number one. We established a whole platform that takes the wild type CRISPR nucleases and engineers them in all sorts of ways that we have developed. And of course, we use AI for that. We use my personal and others in the company personal background in protein engineering in order to get that. I can say, fast forwarding to our lead program, I think that we're probably the first ones that were able to present the FDA with actual data that shows non-detectable off-target whatsoever.
Wow.
Even at some point we had just one, and then we had to deal with the fact, and we made it go away, and then we had to deal with the fact that we had to explain the FDA that we had one, but now this one is gone as well. So and actually it is a challenge when you have to explain such thing. So that was objective one, number one. Objective one, number two, again, in order to make the technology available to more and more patients, is make more genes targetable.
Mm,
How do you do that? So there are a couple of ways to do that. One thing that we chose is that we said, okay, there's no one solution fits all in biology. The nucleus that was originally discovered in 2012 is SPCas9, and it's a great nucleus. It's not the only one. There are thousands and thousands of nucleus, and they're different from one another, have different sizes, they have different functionalities. So we said, okay, so first of all, in order to broaden the potential applications of CRISPR, we don't want to rely just on one. We want superior nucleases that are different from one another. Based on that, we are able to apply additional editing strategies. So we're able to target more loci eventually along the genome. Just to give you an idea, with SPCas9 alone, you target more or less 5% of sites along the genome. With our panel of nucleases, we cover 86%. It gives you flexibility
sure.
eventually.
Yeah.
And in addition to that, we were able to develop novel editing strategies, and we were working on several effectors that are conjugated to the CRISPR nucleases in order to make our toolbox as rich as possible. And with all of that combined with unprecedented safety or off-target profile, I think we will be able to treat more diseases eventually.
That's amazing. I mean, it was actually one of my questions for you. Just thinking about today's episode, I was wondering, is Cas9 the only, the only way to edit? But you've answered that. You've answered that question very well.
Mm-hmm.
That you guys are actually developing, other methods. And then I picked up on your website as well. For anybody that's not aware of this, on your website it says that Emendo wants to make CRISPR better, and that's where a lot of this description, as well, is coming from. I believe.
Yeah, exactly from that. Like trying to make CRISPR better eventually is CRISPR is a holistic technology. And you see, by the way, gene editing, I think, there are, of course, there are a couple of gene editing companies out there, a couple of CRISPR gene editing companies out there, and more companies. There are developing, again, different editing tools. And eventually, combine together all of these technologies, I think that they really have the potential of changing a lot of what of healthcare as we know it today.
Yeah, well, I mean, I just think how far we've come, just in general with other modalities, and now with the promise of what CRISPR could deliver as well. I'm looking forward to seeing the impact that it can have on mankind and the diseases that we face today. In terms of the pipeline that you're working on at Emendo, Can you give us any examples of something that we might see sooner rather than later?
Sure. So our lead indication targets severe congenital neutropenia, or ELANE. The gene is ELANE. Uh, it's and I'll relate the disease to Emendo's platform technology. I have mentioned at the very beginning that we have developed tools that gets us to extreme levels of specificity, meaning eliminating off target altogether. But when you have highly specific or extremely specific editing tools, what can you do with that others cannot? And then we
Mm-hmm.
thought, okay, so we're eliminating off target. Just to give you an idea, off target, you have the on target site, right? That's where you want to cut. Off target usually would have on average, three mismatches along the genome. CRISPR targets, let's say, between 18 to 22 bases. So you'll have three mismatches, and that means you get a certain level of cuts where you don't want it to happen.
Mm-hmm.
We said, okay, but we were able to eliminate that. But then we thought that we want to actually make more genes targetable, right? We don't want to go after the same exact targets as everyone else goes after. And then we say, okay, what if we could do allele specific editing in a broad manner? You have two alleles, one you get from your mother, the other from your father, and they have very little differences between them, right?
Right.
But these differences are either mutations or SNPs that are different. So we developed a technology that, in a broad manner, can differentiate between the two alleles based on a single mismatch, and it doesn't matter to us where the mismatch is. ELANE is an autosomal dominant disease or a dominant negative disease. That means that you have one allele that carries a mutation that generates a toxic effect, and you have another allele that produces a healthy protein that you want to keep. If you're able to do allele-specific editing, you're able to leave the healthy allele intact to keep producing the healthy protein that is needed, and you manipulate only the mutated allele. So that's ELANE. We were able to do exactly that. So we're the first ones who are developing in advanced stages in a little specific approach. We will file our IND Q4 this year, and then Q1 24. We're excited to start, with the efforts to cure these patients.
Amazing. Yeah,
Yeah, and that's
absolutely.
the first indication. Um, I would love to tell you more about our pipeline, because it also has a kind of I don't know, methodology behind it,
Yeah,
because we had, yeah.
absolutely. Please share. Yes, I mean, and I do want to encourage anybody that's listening to this as well to go visit the Emendo website, because you have your pipeline, out there as well, that I saw as I was prepping for the show. And I mean, it's amazing to see how many things that you have lined up. I mean, it's not just one thing; it's several. So I'll let you continue on, though.
Right. Yeah, we're working on quite a few things these days. But again, if I go back to our motivation at the very beginning, we said that eventually we want to treat as many diseases, as many target genes, and as many people, right?
Mm.
So we have started for very obvious reasons with ELANE, which is a rare genetic disorder. There is a very a clear connection between the genotype and the phenotype eventually. And it's a very good starting point. And I think the entire industry started with that, right? You want to treat the severe diseases that has a clear connection between genotype and phenotype. Going forward, we said, okay, but we want, of course, to continue treating rare diseases, but what about more prevalent diseases? So we kind of designed our pipeline in a way that will first validate the technology by treating severe rare diseases, going more and more towards more prevalent and potentially non-monogenic diseases. And I think that that's the promise of CRISPR gene editing or gene editing generally. And I think that that's where the entire industry is heading for. And the more companies that generate data, and there are quite a few, and data accumulates, and we see that they actually, we get more confidence about safety and about efficacy, then it will for sure move to that direction. So at Emendo, we decided we'll target cardiovascular diseases. That's our next priority in the pipeline. And we have chose LDLR, the LDL receptor, as the target, and ANGPTL3 as our next targets. Now, for those of you who are not familiar with hypercholesterolemia or familial hypercholesterolemia, that's our next indication. It affects, it is a monogenic disease, but it affects 1 in 220 people worldwide. So it's highly prevalent. The treatments that are available for these patients are statins that we all know, right? Daily. So that's one thing. And there are the new kids in the block are anti-PCSK9s or PCSK9 inhibitors. And then under development, there's for instance Verve, who knocks out PCSK9 altogether. Why statins and PCSK9, and what's different about Emendo? So statins and PCSK9, both of them are aiming for the same result. That is, increasing the level of LDL receptors that are presented on the surface of the hepatocytes. And when you have more receptors, you increase the uptake of LDL from the bloodstream, and therefore you have lower levels of LDL, which is proven to be an important thing. So the thing is that even though you have these existing solutions, you have, I think today, more than 7 million people just in the US and EU that are not at goal, although they take both. So something is missing there. And we thought that from the very beginning, let's see if we can find with the Emendo's platform, with our ability to generate this or to develop this novel editing strategy, something that will be more efficient than that. And we have developed a new editing strategy that, interestingly, we took that principle we took from a paper that we saw that was showing an Icelandic family. And this specific Icelandic family had a deletion of part of the 3' UTR, which is part of the LDLR gene. And the outcome of this natural deletion was that they had a significant elevation of the representation of LDLR. They had very low levels of LDL, and they were generally very healthy. So we said, okay, that's interesting. It works direct on the LDLR gene, and it shows very promising. It seems like a very promising approach. So we took the natural thing, and of course, optimized it, made it better. The deletion that we have gotten to is significantly more effective. And what we would want to offer eventually is the one and done treatment to hypercholesterolemia. We'll start with familial hypercholesterolemia, which is, again, monogenic form. But there, as I said, there are many, many people out there that are not at goal, although they take both treatments, and I think it's a huge promise. By the way, not just for the patients or the themselves, but generally for the public. Like
Right.
if you, if you, it's that's why I think, if you do that and it works really, really well. And by the way, you have to price it properly eventually in order to get to as many people, because as many people as possible, and that's a whole other discussion. But if you're able to do that, you will eventually get a significant decrease in mortality due to cardiovascular diseases, which is still the number one cause of death in many countries.
Right.
So I think that's actually I'm looking forward
Yeah, very
to see that.
promising. Yeah,
Yeah.
I was actually, it was interesting to me. So I was actually in the best shape of my life, and my cholesterol was high enough that I needed statins, and slowly changed my diet around enough that I no longer needed statins anymore. So I'm still not on them, and each time they test my blood, it comes back fine. But it was one of those moments in life where I remember telling the doctor, I was like, look, I mean, I'm carrying around the least amount of fat that I've had on my body in my entire life. And yet you're saying that my cholesterol is high. And so we did, I think it was 2 or 3 rounds of, testing along the way. And it did come back under control, with statins pretty quickly. But then, my whole goal then was how do I find my way back off of these, because I didn't want to be on
Hmm.
them, and so I got things under control. But yeah, I do look forward to seeing more solutions like this in our future, because I think similar to my story, other people may feel healthy or be at their healthiest point, and then just have something that isn't quite right. And wouldn't it be nice to just have a one and done solution, to your point? That would be wonderful. In your career, what is one of the biggest challenges that you've faced, and what did you learn from it?
Um, well, I had quite a few. It's uh, 25 years of a career at the moment, so I had quite a few. But uh, well, I'll first speak generally about the biggest challenge that I see in what I do, and that's you know when you're the CEO of a company, uh, you have to manage many different interests and wills uh, of different, really different people, different motivations, different interests. You have investors on one hand, and they want they want their money back at least, and preferably to make significant earnings on their investments. You have that. You have the people that work really hard with you in order to develop this the technology and the great solution and the promise. And above all that, you have the patience.
Hmm.
Uh, com- you know, Considering all of these different interests is sometimes really, really hard. I had experienced that specific thing when I was, when we had the discussions or the negotiations back in 2020, or whether to merge or whether not to merge, and you have really different interests around the table, around the same table, and you have to make decisions. If you go one way, someone will be unhappy with that. If you go the other way, Another one will be unhappy with that. But eventually, you're in a position where you have to, and that's the preferable option, when you're able to get everyone aligned, understanding that no one gets everything that they expect, but, and that's something that I strongly believe in, the best solution or the best option is the one that makes everyone happy to some extent. Everyone has to give up something, they get something in return, and then and from that point you move forward. When it's possible, that's what you want to get. Sometimes it is not possible, and you still have to make a decision because you have a company eventually that you need to lead, and but I found that to be the most significant challenge. Also, think about financially. You have investors on one hand; they want to see results very fast. They want to see value creation. You have patience. You want, and I said from the very beginning, the first motivation that we had, first goal was to create an unprecedented level of safety, but it takes time to develop it. So
Yeah.
what is the sweet spot there that gets you to a point where you say, okay, this is enough. Now we have to move forward. This is it. This is design freeze. From this point on, we go forward. So that's just some of the challenges.
Yeah, I would say, I mean, being the leader of a company like Emendo as well is can't be easy at all times, but at the same time rewarding, hopefully on the back end as well, as you start to see the impact that it's having on patients. So, yeah, I definitely can appreciate that position as being a challenge and a way to learn along the way. How do I evaluate my trade-offs? And, at the end of the day, nothing's going to be 100% accepted by everybody. That's just the way that the world works. So I agree.
True. By the way, I'll just add to that the only ones that I think that you cannot compromise and their interests are eventually the patients.
Yes. Yes. Yeah.
There, you cannot make any compromises.
Very important point. So there are 3 questions that I like to ask every guest. What inspires you?
Um, I think that I'm inspired by uh, human beings that make significant impact on the world when they have the right positive motivation to do so. So it's not only what you do. I think that I think that almost a motivation what motivates you is more important than the outcome in many cases. And I will forgive someone who had the right positive motivation that wanted to do good, but eventually things turn around the wrong way, than someone who had the wrong motivation and somehow managed to do something that is amazing. Although yes, especially as an entrepreneur, as a CEO, I do believe that execution and getting what you intended to get is eventually, that's how you're measured. That's how everything is measured, and that's my everyday. But I don't miss out on the motivation part. And I think that's there are wonderful human beings out there doing wonderful things. But I would always look at what motivates them as something that is a very, very significant and important element.
So important. Thank you for sharing that. And what concerns you?
Where should I start? Well, I'll say, I think that what concerns me at the moment is something that concerns all of us. So I won't be original here, but I know AI very well. I have a very strong computational team. We use AI. We develop deep learning algorithms in house. We use them. We enjoy the outcomes. The more you know AI, uh, the more you realize uh, the potential potential great things that you can do it, but how devastating it could be if it is not controlled.
Hmm.
So, I love technology. I love what we do at the Mendo, but I think that this is the first time. Once I got to the idea that got used to the idea that we're going manipulating people's genes, and at the beginning it took me a while. uh, I think that this is tech wise, or if even if you look at all developments in the past few decades, this is the scariest thing, the most wonderful thing that happened to us, but also the scariest ones. And I really, really hope that regulators, governments, whoever is there, I hope they work really hard to define a framework for AI developers, and boundaries and controls, uh, because you can do good with it, and you could potentially do really bad with it.
Yeah.
And
Yeah.
that's, I'm telling you, as someone who practically develops AI technologies, and someone who enjoys that a lot. It saves us a lot of money and time.
Hmm.
But there's a big but over there. Where do
Right.
you stop it?
Yeah, I think that's kind of everybody's fear, and then I know from my side with the U.S. government wanting to, sort of understand it more and like take a step in. As much as I appreciate that, whenever it came to the understanding the internet, initially they were describing the internet as a series of pipes that joins all of our houses together, and it's like, okay, come on, guys, you got to come up to like at least where we are today, and then gain an understanding, and then you can, maybe build a task force or something like that to go, look at ways to better regulate this, right? Because I agree with you. I mean, it's both exciting and scary at the same time. Last question: what excites you?
um, what excites me? Um, well, I think that with that, I think that achieving uh, my goals, that excites me. Setting goals, and then just seeing it happening
Hmm.
real,
Yeah.
especially as an entrepreneur, especially as an inventor. Like I have, I've written many patents, I've founded companies, and when you think about it, and you say, okay, just a moment, this is something that uh, if I wouldn't have done it, or if wouldn't have done it, it wouldn't have been here. Looking at creations like that, and again, I go back to the motivation thing. I hope that I come, that I come with the right motivation, seeing it materialize, seeing it becoming something that wouldn't have existed otherwise. The realization, that's very satisfying and exciting. It goes away after 24 hours, and then you and then you go after the next goal. But
Right.
still, that's one of my biggest drivers.
Amazing. Well, David Baram, thank you so much for sharing all of your thoughts with us today and talking to us about Emendo and all of the great work that you guys are doing. And thanks a lot for being on the Life Science Success Podcast.
Thank you, Don. It was a pleasure. It was